Okay, freshmen, good morning. Happy Friday. Congratulations for making it through your first week of quarantined online virtual learning. Hopefully it hasn't been too confusing and too much of a headache, but I know it's going to be a struggle for you guys. There's no way around it. It's a frustrating experience for everybody, but again, staying safe, staying calm, getting through it is most important. But anyways, I have on this video the next 2 days of class. I think a YouTube video will be easier to just click the link and stream instead of downloading. But same deal as always, personal questions, send me an email, content or other issues, you can post to Edmodo and use me and your peers to help you. And then lastly, next Thursday, I'll try to set up a live Zoom so students can check in with other questions about bio and just to kind of see each other, see how everybody's doing and get a little bit of feedback. So anyways, today's our first day of genetics. So we're calling it day one of the genetics unit. It's Friday, March 20th. I don't think we're going to do bell ringers for this unit. I think it's just one more thing to be confused on in terms of formatting, whatever. So let's just focus on the big picture on this, the big ideas. The unit will be about 6 class periods, assuming we come back after spring break, and the days will be pretty straightforward where I'll have a lecture and a homework assignment. Obviously, you're doing it all at home. The lecture should not be more than 15 or 20 minutes. Some days will be even shorter. And then the homework, I think, will be another maybe 20 minutes. So I think in terms of the amount of work, all these classes should be less than an hour. So anyways, let's get out our notebooks and we'll get started with the first notes for genetics. This is our 7th topic, so feel free to pause the video now to get your binder organized and set up. I know you guys need like 7 to 12 different colors for some of you, so get all those colors going. Again, this should be a YouTube video, so you guys can pause, go backwards, go forward, whatever works best for you to get through this. But anyways, here we go. First thing I want you guys to understand is the difference between heredity and genetics. Feel free to pause the video now and just do a little bit of brainstorming. You've probably heard both these terms before. You've probably never thought about the differences between them. So go ahead and brainstorm now. And I know you and your parents are probably already starting to get bored of each other. So next time they're on your case about whatever you're doing, you can remind them that you are the way that you are because of heredity, because of genetics. But anyways, heredity is simply the transfer of genetic traits from one parent to the offspring. It's just the reason why you look like your parents, you often act like your parents, you have similar traits as your parents. But the scientific study of this aspect is a newer science, and that's known as genetics. Scientists have known about heredity, well, really people have known about heredity for, thousands of years. We've understood that offspring look like their parents, but the scientific study of genetics is newer. It's not as new as DNA, which we discovered in the 1950s, but we are going to start with the discovery of this idea of genetics, and that goes back to the 1800s to Gregor Mendel. He was a monk who's considered the father of genetics. He was really the first person to discover that parents contribute equal amounts of genes to offspring, but he disproved the older blending theory, which was what scientists thought before then. This is a picture of his study species, which you may have remembered learning about in middle school, which was peas, and there are some specific reasons why he chose peas to study. Mendel worked with garden peas, which was an ideal species to study for a few different reasons. First of all, they're hermaphroditic, meaning they can self-breed. They have both male and female anatomy. And by self-breeding species, you can basically make a control group. You can isolate the self population. They reproduce quickly with lots of offspring. Think about if you're dealing with something like humans, we reproduce so slowly and we have one baby at a time, sometimes two babies at a time, but still it's hard to get a good idea of traits when there's so much luck involved with humans. But when you have 500 or 1,000 offspring at a time, it really turns a anecdotal finding into, statistically significance. And then lastly, they have really simple traits. So think about humans, we obviously come in hundreds of different sizes and shapes, and peas are really just tall or short, or they're purple or white. There's no in between, and so they have these very binary, simplistic traits. So he started with purple and white flowers, and they were true breeding, meaning when they reproduce with themselves, they only produce offspring that look exactly like them. You've probably heard this term before, like a purebred dog, which is 100% golden retriever or 100% Rottweiler. Similar, where these are true breeding purple and true breeding white pea pods. He called them the parental generation, so this would be P1 is what he labeled them. And he bred them. What that means is he took the pollen or the sperm from one of them and mixed it with the ovule or the egg of the other one. And then he had lots of pea pod babies. What I want you to do right now in your notes is try your best to answer these questions. So taking pure purple and pure white offspring, pure purple and pure white parents. What do you think the offspring looked like? Like if they had 100 babies, would they all be purple, all be white, half and half? Would they be purple with white spots? Whatever, predictions you guys have. And then come up with a reason for your prediction. And maybe this stems from what you already learned back in middle school. Okay, once you've made your predictions, let's go ahead and look at what actually happened. So he crossed the white and the purple, the P1 generation. He called the offspring the first filial generation. He labeled them F1. And they were, dun 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 dun, all purple. And that might be surprising to some of you. That might not be surprising to others. But basically what this meant is that whatever DNA or whatever genetics was in the purple parents seemed to overshadow or overpower the DNA or the genetics from the white flower. But here's where it gets really interesting. He then self-bred the offspring. So basically he had the purple offspring breed with themselves or with each other. I know that seems gross if we're thinking of humans and animals, but with plants, it's really not, inbreeding is not a taboo in the plant world. But here's what's interesting. You need to realize that these offspring, although they're purple, they are not purebred purple because they have a mixture of DNA from the parents. They do have DNA from the white flowers, making them non-purebred. So he self-pollinated the F1 generation. What do you think those offspring looked like? The F1 generation pollinating itself. Turns out that there is a pretty clear ratio of 75% purple and 75% purple and 25% white, meaning a 3-to-1 ratio. And so this all led to Mendel's theory. And so here's where I want you guys to jot this down in your notes. He took these different observations and put together a few set of inferences that led to his theory. But basically, for every trait, an individual has two versions of the gene. Meaning, and you guys know this from homologous chromosomes, we get one copy from mom and one copy from dad. And he called these alleles. The alleles being the whatever version of the gene it may be. So the alleles for, say, height in a pea pod would be tall and short. The alleles for color would be purple and white. Just explained that. He understood that these alleles can be dominant or recessive. In the example I just gave you, obviously the purple allele is dominant over the recessive, over the white. He came up with some terms. These, I'd say, is the most difficult vocab word to start this unit, is understanding the difference between homozygous and heterozygous. Homozygous, and you guys should know using your Latin clues that homo meaning same. This is when you have same alleles for a trait, and so an individual could be homozygous dominant or homozygous recessive if they had two dominant alleles, meaning two of the same dominant alleles, or two of the same recessive alleles. Or an individual could be heterozygous, meaning they have different alleles for a trait. Hetero being Latin for different or other. So again, here's an example of homozygous alleles on a pair of chromosomes. Here's an example of heterozygous alleles on the pair of homologous chromosomes. And again, this could be hair color or height or eye color, something like that. Mendel came up with this idea of genotype and phenotype. So genotype is the pair of alleles for a trait. I remember genotype as being the genes. So this is not necessarily what you see on the outside, but this is the genetic makeup for that trait. And then phenotype is the physical trait caused by the genotype. I remember this with the pH and phenotype being for physical, like this is what you actually see. And then just connecting this back to the cell cycle and what we learned about DNA and cell cycle last unit, you get one allele per trait from each parent. Meaning if you're heterozygous, the dominant allele gets expressed because, as we just learned, it overpowers the recessive. So Mendel then developed two laws, which you've already learned about. The law of segregation, that the members of each pair of alleles separate when gametes form, and the law of independent assortment. Pairs of alleles separate independently from each other. That happens in meiosis 1 metaphase 1 of meiosis, when there's no rule that says which homologous pairs line up on which side. Some of the rules of simple inheritance that Mendel came up with, you might want to jot this down if this stuff is new to you, but I do have it in red because I know some of you have learned this stuff. So this we already know. One allele, whether it's dominant or recessive, is inherited from each parent. We pick any letter to represent that trait. The letter you pick is, it really doesn't matter. You just pick a letter, and then if it's a dominant trait, you write it in capital letters, and if it's a recessive trait, you write it in lowercase letters. So if we were talking about trait A, and trait A could be for widow's peak or a curled tongue or curly hair, could be for anything. Capital A is considered homozygous dominant. Capital A lowercase a is heterozygous. Lowercase is homozygous recessive. So here's the terminology and what we've talked about so far. If we have a capital P for purple and a lowercase p for white, and we showed in Mendel's experiment that the purple flower was dominant to the white flower. Capital P would be a purple flower, and we would call it homozygous dominant. Lowercase would be a white flower, and it would be called homozygous recessive. And then here's the part that is, trickiest or a little bit different from what we might just infer without knowing genetics. Capital P, lowercase p, meaning the flower has one gene for the dominant trait and one gene for the recessive trait. It would be a purple flower because purple dominates over white, but we will call it heterozygous. And so in this example, let me try to star this for you guys. This and this, we would say, have the same phenotype but different genotypes. And when I say the same phenotype, I mean the physical characteristics are the same. They're both purple. Different genotype because the genetics of the two are different. They have different alleles. Okay, and this brings us to Punnett squares, which I know a lot of you are familiar with. One thing I want to remind you is that Punnett squares are for predicting, that's a bad color, predicting the offspring outcomes. It's not showing you what actually happened. It's just showing you the likelihood of future events. And that's something that's important in our terminology and when we get into answering questions later on in the unit. We write genotypes as a ratio of how many homozygous dominants to how many heterozygous to how many homozygous recessives. And phenotype gets written as a ratio of how many dominant to how many recessive. The biggest thing I think you need to remember is that for genotype, there are 3 outcomes, and for phenotype, there's 2 outcomes, and we generally write them, or we do write them in order from dominant to recessive. So for phenotype, it's pretty obvious. We write dominant colon recessive. But for genotype, we write how many dominant, colon, how many heterozygous, colon, how many recessive. And if this doesn't make sense to you, you can always go backwards, but I think when we start doing practice, it'll look clearer. Okay, so let's go backwards and look at Mendel's first experiment when he bred those pure breeding purple and white flowers. So the genotypes of the offspring between a P1 cross would look like, we would have the purple plant, which is capital P on one side of the Punnett square. It doesn't matter. I could have put it over here or over here. And then I'm going to have the white plant, which is little p, over on the other side. And then in terms of the cross, all we do is we take the letters and we put them in the boxes below. And then we take these letters and we put them in the boxes over to the right. And so if I draw this out, it'll look like this, 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 and this. I apologize for my bad handwriting. Notice that I exaggerate the size because a lowercase and uppercase p look the same. So on a worksheet or on a quiz, I would definitely want to make it obvious what a capital letter is and what a lowercase letter is. Again, if we put the purple plant on the left side and the white plant on the top, our result will still be the same. So if I cross, I get P, big P's and little p's. All of the offspring are considered heterozygous. So the cross is this. The genotype ratio is this. And the phenotype ratio is that. Because if we look at the genotype ratio, how many homozygous dominant do we have? 0. How many heterozygous do we have? 4. How many homozygous recessive do we have? 0. If we look at the phenotype ratio, how many dominant? 4. How many recessive? 0. Now if we look at Mendel's second experiment, this is when he crossed the F1 offspring generation with themselves, and remember he got that 75% ratio of purple compared to 25% white. And again, we can look mathematically at the probability that led to that. So here, both parents, meaning the F1, had the same genotype. They were both heterozygous, so it doesn't matter who goes on which side. And then when we cross these, this box is going to be homozygous dominant. These two boxes are going to be heterozygous. And then this last box is going to be homozygous recessive. So the cross is heterozygous and heterozygous. The genotype ratio is 1:1, and the phenotype ratio is 3:1. Now again, if these pea pods had 4 babies and there was 3 that were purple and 1 that was white, that wouldn't really teach us a whole lot, because that could be because of just luck or probability. Think about it. Just because the chances of having a boy or girl is 50-50 doesn't mean that every family with 2 kids has 1 girl and 1 boy. But because he used pea pods, they had thousands of babies, and on average it was pretty much 75% purple and 25% white. And it's that phenotype ratio, because remember, we can't see the genotype. The genotype is the genes, and Mendel was not able to see the genetics here. He was able just to see the phenotype and then infer the genotype and infer how that was solved. Normally in class, I would poll you guys to see how many of you can do these different things. I can share mine, and you can obviously do this individually. But all these different traits, like one of them is a tongue roll, which is this. I can do that. Some of you probably can, some of you can't. Another is free earlobes. Free earlobes is that there's no like skin attaching your earlobe to your head. I don't have free earlobes. Some of you probably do. A widow's peak. That is a like what Dracula has, like a peak or a tip that goes right there at your hairline. I don't have that. A cleft chin. I don't have that. Hand clasp. This one's pretty interesting. Just take your hands, don't think about it, and put them over each other, and then look at which thumb is on top. And for me, my right goes over my left, so I don't have that trait. And then lastly, 6 fingers. I also don't have that. And so the only reason why I'm asking these different things is because there's this misconception that I want you guys to understand. And that is that dominant traits are not always common. Just because a trait overpowers another one, like just because purple overpowers white with the pea pods, it does not always mean that there's more purple plants. It could be a very rare trait. It could be that the majority of a population, like with 6 fingers, I know for a fact the majority of my students do not have that trait. Even though that's a dominant trait, we pretty much all have the recessive trait, which is to have 5 fingers. So again, all I want you to get down from this is that dominance is not indicative of commonality. Interesting example of this is Huntington's disease, which is a rare disease that's caused by a dominant allele. Most diseases are caused by recessive alleles. Take a second here and brainstorm, why would it be rare that a disease is caused by a dominant allele? In other words, why are most diseases recessive? Think about that question, because it's actually going to come up later in the unit when we learn about diseases. So, we can look at Punnett squares to come up with the answers to questions like this. What is the probability that this couple will have a child with Huntington's disease? Remember, Huntington's disease is a dominant trait. Since we're using H, this man is heterozygous, meaning he does have Huntington's disease. This woman is homozygous recessive, meaning she does not have Huntington's disease. I don't know why I can't draw an X right now. Oh well. But anyways, try your best to solve this. Pause the video, make a Punnett square, and try to answer this question. All right, well, the man's genotype would look like this. The woman's genotype would look like that, meaning the offspring would have a 50% chance to have Huntington's and a 50% chance to not have Huntington's. So the answer there would be C, 1 and 2. But again, realize that this is just probability. This couple could have 6 kids and they could all not have Huntington's. Or they could have 3 kids and all 3 could have Huntington's. This is just probability. Think about flipping a coin. You know that a probability of flipping a coin is 50% heads, 50% tails. But I'm sure you could flip a coin and get 4 heads in a row or 7 tails in a row. It's just that the odds are 50% will be heads and 50% will be tails, similar to this question. Okay, guys, so that is it for today's lecture, the introduction to genetics and Punnett squares. Like always, please advocate for your own learning. So go online, Google Punnett square tutorial, go to YouTube and watch videos on how to do Punnett squares if you need more practice. Obviously, you can go back and rewatch any part of this video. But hopefully it all made sense to you. Your homework for the weekend is the Punnett square packet, pages 1 and 2. It's a 10-page packet. Go ahead and print it all out now, staple it together, but you're just responsible to complete the first 2 pages, and then we'll check that in on Tuesday's video. And remember, we will plan to do a Zoom meeting on Thursday during class time. I can send out information on that on Tuesday's video. Okay, hope everyone's doing okay and have a good weekend.